Hey guys, Mr. P. In this video we're going to talk about mitosis, specifically IB understandings D217 and D218. D217 talks about the phases of mitosis and expects students to know the names of the phases and how the process as a whole produces two genetically identical daughter cells. And the video will end with D218, which is all about identification of phases of mitosis. So this is a skill that requires you to use diagrams or view actual microscope slides or micrographs, which are pictures of microscope slides, in order to identify phases within the tissue sample. So the first thing we need to do is identify the phases of mitosis. And so as you can see on the screen, the phases would be prophase, metaphase, anaphase, and telophase. Those are the four phases. We typically use the acronym PMAT, prophase, metaphase, anaphase, telophase, in order to remember the four phases of mitosis. Remember, mitosis is by definition the division of the nucleus, so we are not technically referring to cell division as a whole. Mitosis rather is dividing the nucleus of the cell into two identical daughter nuclei before cytokinesis takes place, which is by definition the division of the cytoplasm. So if we focus on prophase to begin our discussion of mitosis, the first thing that is indicative of mitotic prophase is that the chromatin fibers become more tightly coiled to form chromosomes. In the last video, we talked all about DNA condensation. We talked about the how and why DNA condenses. But in interphase, which is the phase preceding prophase, all of the DNA is kept in that inactive form, which is called chromatin. During prophase, the first thing that happens is the chromatin, which is the inactive form of DNA, has to condense in order to form the chromosomes, which becomes the active form of DNA. We have to constrict it into usable and manageable sized chunks called chromosomes. The nuclear envelope, which is this nuclear membrane that surrounds the nucleus, is going to disintegrate and the nucleoli disappears. So as you can see, we have just standalone chromosomes. A mitotic spindle forms as the centrosome builds new microtubules that will be used to pull the chromosomes into positions. These are centrioles, and those centrioles are going to begin to form those microtubules. This entire thing becomes the mitotic spindle. It is also called the centrosome, and the centrosome is going to be instrumental in not only moving the chromosomes around, but also dividing the chromosomes and then producing the elongated cell that is indicative of anaphase. The kinetochores, which are a region in the centromere of each chromosome, attaches to the spindles. So if we were to blow up this structure, our chromosomes actually look like this. There is a centromere here, which holds the two sister chromatids together, and on the centromere are two little proteins called kinetochores. Those kinetochores are going to be the protein complex of the centromere that allows that microtubule to attach to the centromere. And the centrosomes move towards the opposite poles of the cell as a result of the lengthening of these microtubules. So as these microtubules lengthen, especially when they connect to each other, they're going to allow these centrioles and centrosomes to continue moving further into the poles, which, as you can see, starts to lengthen the cell. Metaphase is characterized by the chromosomes moving to the middle or the equator of the cell, which is called the metaphase plate. Metaphase plate is this kind of imaginary line that runs right through the equator of the cell. These chromosomes are going to line up one on top of each other until we make a line of chromosomes. The centromeres of the centrosome align on this plate. You can see that the centromere is completely lined up and is located exactly on that metaphase plate. The chromosomes move as a result of the action of the spindle, which is made of microtubules. So you'll see that some of these microtubules will attach to the kinetochore and will therefore be instrumental in pulling these chromosomes apart. However, some of the microtubules actually bypass the chromosomes and will connect to each other. And if you were to zoom in, they actually don't fully connect. They actually overlap in the middle, and that's what those motor proteins we're going to be bound to in order to move these microtubules in opposite directions, which leads to lengthening of the cell, which leads to cell elongation. The centrosomes are at opposite poles. They have ended their migration. Prophase, they obviously were migrating. In metaphase, they ended their migration. The centrosome is completely set. The spindle apparatus is completely set. All of the chromosomes have lined up, and the microtubules have completely attached to all of the kinetochores of the centromeres, and we are ready for anaphase. In anaphase, the chromatids, now each a chromosome, move towards the opposite poles of the cell. You can see that in metaphase, all of the chromosomes lined up, and anaphase is characterized by the chromatids being pulled apart. In mitotic anaphase, those chromatids were separated and are moving towards their poles, or moving towards their centrosomes. All of these microtubules will be shortening, which are being pulled in tightly to the centrosome, which is going to eventually become the new nuclei. They move as a result of motor proteins, again pushing those microtubules in opposite directions. And the centromeres are attached to the microtubules. They move towards the poles first. At the end of this phase, each pole of the cell has a complete identical set of the chromosomes, and that will eventually lead to the new nuclei forming. Telophase is characterized by the new nuclei forming, and so the first thing we would need to do is we would need to get all of the chromosomes to each of the poles. Once they end their migration and they have moved away from each other and away from that metaphase plate, they would be located in each pole. We then form a new nuclear membrane around them. The chromosomes start to elongate, the nucleoli reappear, the spindle apparatus disappears, and the cell is lengthened and fully elongated and is now ready for cytokinesis, which, as you recall, cytokinesis is the division of the cytoplasm. If we look at this cell all the way on the right, this is called the cleavage furrow. And the cleavage furrow is a pinching in of the cell membrane on both sides until it eventually pinches in enough to where the membranes touch. At that point, it completely snaps the membranes apart, and these become the two new daughter cells, which will again go through their own cell cycle, starting with G1. D218, again, was the identification of phases of mitosis, and so when you look at micrographs like this particular image, you will see a variety of cells. Every single one of these little squares would be a cell. You can see the nuclei within the cell. These are the dark circles within the cell. If we isolate what the nuclei should look like for the identification of phases, which is indicative of this smaller image, you will see that interphase is typically associated with a really uniform dark nucleus. Prophase, you begin to see the chromosomes condense and form individual structures. Metaphase, the chromosomes line up in the middle. Anaphase, the chromatids have been pulled apart, so there are two separate groups of chromatids. And then in telophase, you typically start to see that new cell wall forming, and the new nucleus is going to be more intact than they were in anaphase, which is the previous phase. So when we look for these particular phases, both interphase and M phase, which is characterized by P, M, A, and T, on a micrograph, such as the image on the left, you begin to see a bunch of cells that kind of stand out, which I highlighted in these boxes. And when you further investigate the cells within those red boxes, you see that there are a variety of cells in both interphase and mitosis. And then of the cells that are in mitosis, we have cells that are in prophase, metaphase, anaphase, and telophase, and we can associate or label all of those phases here. Again, prophase, we see individual chromosomes, which are kind of becoming more individualized from these types of nuclei or these types of cells that would be indicative of interphase. You see metaphase, which is the chromosomes lining up in the middle. You see anaphase, which are the chromatids beginning to separate, and then you see telophase, which is full separation and new cell walls forming between them. That's it for this particular video. If you learned something, give it a thumbs up, subscribe to the channel, leave any questions in the comments. We'll see you later.